Okay, so I will talk a bit about an ongoing project of us in which we focus on defects in membranes. This project has been going on for a little over 1 year, which, yeah, thanks to COVID, has been suffering from significant delays as well. So I am Arno Daze. I'm a postdoc at Paint group in UGent, and our group consists basically of 2 part-time professors, Arne Verliefden and Emil Cornelissen. Emil is also affiliated to KWR, and we have currently 4 postdocs working with us. So besides me, there's Marjolein van Oppen, which I guess many of you know, then Ivailo Hitsov and Leonardo Gutierrez. Our group focuses on physicochemical water treatment, and this is all for drinking water as well as for processed water. The technologies that we focus on are mainly membrane-based, so reverse osmosis, electrodialysis, membrane distillation, and forward osmosis, but we also do some ion exchange and coagulation flocculation tests. And another important leg in our research is quality of boiler water and how the high temperature influences water composition and corrosion of the boiler equipment. But I'm not involved in the boiler part. So in our project here, we will focus on defects in dense membranes. So dense membranes, that would be RO and FO membranes mainly. That we focus on. So as you all know, those membranes have a porous support with a top layer which should be non-porous. On the left, you can see what the active layer should be doing. So it permits water flow while rejecting solutes. But then if there's a defect, we get a much larger flow rate through that defect, and with it also solutes that can pass into the permeate sites. Now, is this actually a problem that there are some defects in membranes? So for RO membranes, for instance, those membranes are already relatively permeable for both water and salts. And so if there are sparse defects, there's actually very little influence of those defects on the permeability and selectivity of the membrane. You can actually tolerate quite a few defects without any noticeable influence on the permeate quality. However, in the case of larger pollutants and pathogens, their defects are basically the only way into the permeate. Viruses, for instance, which have become very relevant in the past year, they are, tens of nanometers to hundreds of nanometers. So an RO membrane without defects should yield total rejection of those pathogens. If they are to be found in the permeate, it is because there are defects in the membrane. Now, whether it's a problem also depends on what the feed of your system is. So the classical application for reverse osmosis is seawater desalination. And in seawater, the concentration of human pathogens and pollutants is fortunately very low. So in fact, in seawater desal, it doesn't really matter that much if there's a small defect in the membrane or not. However, thanks to water scarcity and population growth and so on, we are forced more and more to recycle wastewater, either for drinking water purposes or for processed water purposes. And there, of course, the situation is completely different. If your feed water is of poor quality to begin with, then it really does matter whether pathogens can move through, break through the membrane or not. So in this project, we have the aim of studying quantitatively how defects impact selectivity of membranes, and we are also interested in developing monitoring techniques. And then the overall goal is to end up with an online and real-time membrane integrity monitoring technique, which can serve basically as an early warning that there might be something wrong with a membrane. For this, we want to measure relevant indicators. And as I mentioned, water permeability and salt rejection are in fact not that relevant. They're not sensitive enough. So we need to find better indicators as well. In this project, yeah, so there's me working on it, and besides me, there's Emil Cornelissen who's involved, also through KWR. They're also very interested in membrane integrity monitoring. Then there's Dwani, our PhD student who's working on this project. And then a non-academic partner is Faris, the drinking water company, through Bart de Gusseme and Benjamin Buisgaard. And they're involved because, as you can see on the right, in the city of Ghent, there's the Nieuwe Dokken project, which is going to be a new neighborhood in the port in the north of the city. And there the locally produced domestic wastewater is going to be recycled for process water purposes. So a nearby company which produces detergents is going to buy RO permeate from locally treated wastewater. But of course, they want to be sure that the water that they receive is of high quality. So if we want to study defects quantitatively, we also need to be able to produce defects quantitatively. And this is actually quite difficult to do. So we started by looking at some nano machining and micro machining techniques. A well-known technique, for instance, on the left is focused ion beam, in which gallium metal is ionized and accelerated under very high potential difference, and then those heavy atoms impact the membrane at a certain point, or your sample at a certain point, and transfer their energy to the solid sample, which then starts to eject a bit of the material. Argon gas milling is quite similar. So there, you inject argon gas into a plasma, into a very strong electric field, which creates a plasma, and then again those ions are accelerated and impact your sample, again leading to sputtering of the sample. There are, however, some downsides to both focused ion beam and argon milling, in that you need very high vacuum. So this leads to extremely expensive equipment. And also the sample might not be suitable for filtration afterwards. For instance, argon gas milling relies on a mask to create the pattern that is etched away. And also the samples might be sputter coated to facilitate working on them. So for instance, for the focused ion beam, the samples should be conductive to limit electrical charging. So the samples are sputter coated. So yeah, you can't really use them afterwards if you want to then do filtration tests. So some other techniques that we've been looking into are laser ablation and electrical discharge machining. Now the laser ablation, I've shown here a picture of laser cutting through steel plate. This is of course quite different from what we're doing. The laser ablation is using extremely short laser pulses, really of the picosecond or even femtosecond length to create small bits of plasma, which then etch away a part of the material. We have been able to produce very small and reproducible defects. So the defect that I've shown here is about 4 micron in diameter, but we can go even lower. In theory, we could go down to about 200 nanometers, which is the theoretical lower limits of a deep UV laser. However, laser ablation also suffers from some problems. As you can see here in this array, well, you can see that we can very reproducibly create defects and large numbers of it, unless the surface is uneven. And then, you see within the circled area, you see that the defects are quite a bit smaller than outside of it, and this is due to the surface roughness. So the laser ablation relies on a very small focal plane. So if this is your membrane, so it's a schematic, of course. The active layer is a bit exaggerated here, but basically what you need is a very small focal plane, which is only 2 micrometers high. So above and below this this zone, the laser beam, is not focused enough, does not contain enough energy per surface area to create plasma. So then the membrane is not ablated. But of course, 2 micrometers, it's a very small plane for a membrane. So the membranes, especially at larger scale, are more variable in thickness. So the RO samples that we've been working on so far are usually around 150 micrometers thick, but they're of course not perfectly flat. The laser ablation technique is mainly used in the semiconductor industry, where they work with extremely flat wafers. So for them, our samples are quite challenging. So what we're trying to do now, well, the photonics group that we cooperate with, they are trying to implement some sort of automatic refocusing for each defect. So where the laser would basically find the correct height of the membrane surface area before introducing a defect. And then the second technique that we're now looking into is electrical discharge machining, which, as the name implies, relies on electrical discharges to dose energy. And the advantage here is that we do not suffer from focal plane limitations. So you create a spark across a small gap, and basically everything in between the spark is energized. EDM is used now as a very high precision metal machining technique, as you can see in the GIF that is playing. So this is not some sort of trickery or simulation or something. You can really make parts that are that precise using EDM. However, EDM does not yet exist as a technique to ablate polymeric film, so it's a metalworking technique. I've searched quite a bit for a suitable machine, but I couldn't find one. So we're now prototyping this ourselves. So as you would imagine from a prototype, it looks a bit messy, but this machine can generate pulses of about 5 kilovolts with a minimum duration of a few tenths of microseconds up to as long as you want, basically. So here's a picture of the ablation in progress. So the sample, the white sample, is an RO membrane, is an FT30 membrane. The needle has a width of 100 micrometers. So on the top, the shaft of it is 100 micrometers. The tip is 10 micrometers, and you can see the purple plasma in between where the spark is occurring. And if we then look at those defects under the electron microscope, this is what they look like. They are still quite large so far. And what is also interesting is that the active layer is not always completely breached, or at least it doesn't look fully breached. However, those samples are stained with iron and potassium, so some heavier elements compared to the membrane matrix. And we can see that the stain is penetrating into the membrane at the site of the defect, and it is not penetrating in the surrounding regions. So the active layer is definitely damaged, even though morphologically it might not be entirely obvious. So we need to study further just to what extent the active layer is damaged in these areas.
Arnold, also for you, 5 more minutes, please.
Okay. So the challenge here, as I mentioned, is to create smaller defects still with the electrical discharge machining. So yeah, we need to go to very short pulses to create very small defects, which is work in progress at this point. So if we then introduce defects into membranes and we do some filtration tests with them, this is one of the results that we got. So in this membrane, you can very clearly see the row of defects that we introduced. The row actually extends further with smaller defects still, which are quite hard to see in this picture. This was done with 50 millisecond pulses, with the larger defects consisting of, I believe, 8 pulses, and then smaller defects are consisted of a lower number of 50 millisecond pulses. In the feed, we dosed rhodamine as an indicator and sodium chloride, and it's maybe surprising, but this membrane still displayed more than 90% salt rejection. So even though it is very clearly leaky, it was still rejecting the majority of the salt. Of course, 91% rejection is it's not really suited for RO. So it would make for a very poor RO membrane. But yeah, if you look at this picture, you would expect basically that this membrane wouldn't reject anything anymore. However, what we also saw in our tests, which is also one of the challenges in this project, is that the common lab setups are not always are not exactly suited for this for defects or integrity monitoring. So what we tend to see is that the sealing of the cells causes defects at the edge of the membranes. As you can see here in both pictures, you have this purple ring around the feed flow channel. So this is where the two halves of the flow cell are closed, and they compress the membrane, and they cause some, they cut into the active layer. It is also obvious here in this picture. So this was with a Sterlitech dead-end filtration cell, and you can very clearly see the ring where the O-ring was pressed onto the membrane. And of course, if you press an O-ring onto a membrane, it makes sense that you damage the membrane at that spot. But unfortunately, those damaged areas are still accessible to the feed solution. Otherwise, the rhodamine would not penetrate into the membrane. So ideally, if the cell is closed, the area of membrane that is damaged by the O-ring should also be sealed off, which is not the case. And of course, we've tried many different O-rings and different torques to close the cells and so on, but this seems to be a general problem. It occurs almost every time. And so the filtration performance here of those defected membranes, again, the salt rejection is quite okay, still 99%. So this is with membranes which are basically only damaged at the seals. But then if you look at the fluorescent indicators, you see that the rejection there is quite poor. It is comparable to the sodium chloride rejection. However, those compounds are all very large. They all contain a chromophoric group, so they contain multiple phenyl rings, they contain side chains and so on. So they're quite large compounds, and yet their rejection is between 96 and 99%. You would again expect for a non-defected RO membrane, you would expect a rejection of about 100% really. There's no reason why such large compounds should penetrate through. And also there seems to be no relation between solute size and rejection. So sodium chloride, being a very small solute, is in fact rejected slightly better than the large compounds, which again is not according to theory of an intact membrane. And so the as for the further outlook of this project, so we want to scale up our tests to spiral wound modules. So there we no longer have the issue with cells sealing. If we do find contrast between lab scale setups and spiral wound full scale setups, yeah, this does have a significant impact for lab research, because now if you're doing some sort of benchmarking tests or prototyping tests, you might go to these small kind of setups. But if you're interested in very high rejection of certain problematic compounds, your lab results might be unreliable. So yeah, how do we know whether we can trust our lab setups if we're looking at organic micropollutant removal or API removal or yeah, those kind of compounds? We're also going to extend our tests into virus-based integrity monitoring. So this will be in cooperation with KWR. They've developed a technique based on natural viruses. So basically, if you take any sample of surface water, you will find millions and millions of copies of yeah, viruses, most of them fortunately not pathogenic to us, and you can detect those with PCR. So you can do before and after with just general primers, so for general detection of a certain class of viruses, and this yields very high sensitivity. And then finally, we're also looking into developing flow cytometry as an online tool, as a real-time online tool for RO permeate quality monitoring. And with that, I would like to thank you for your attention.
All right, thank you, Arnaud. I don't see any questions in the chat yet, but maybe I wanted to ask your opinion on the setups you're using in the lab. So you showed that the O-rings produce some damage already on your coupons. Might it be that the staining comes from disassembly? That it only happens then that some residual feed might get in touch with indeed where the O-ring sealed quite tightly on the membrane? Or did you rinse specifically afterwards?
Yeah, we do rinse afterwards, and also the disassembly is relatively fast. So for instance, let me go back a few slides. Oh, here, for instance. So rhodamine is a large compound, so it diffuses quite slowly. So for the rhodamine to penetrate that wide into the membrane, so it's not just going through the active layer and sitting there, it's actually spreading out laterally and vertically throughout the membrane. For this to occur, it needs some time. So if we disassemble, so we flush, we disassemble and flush the coupon again, I mean, it's just seconds that the compounds would have the chance to flow past the feed channel. So we're quite sure that it happens during filtration and not during disassembly.
All right, thank you.
But it's a good point, yeah, that the with this kind, especially if you go to very sensitive techniques, the procedure of sampling and rinsing and so on becomes very important, yeah.
Mm-hmm. And then here your main hypothesis for the 91% rejection of sodium chloride would be?
So the undamaged active layer surrounding the defects is still rejecting salts. So yeah, the flow rate of water through the defects will of course be higher than it is through the undamaged active layer, but the overall contribution of the flow rate through the defects to the total flux is still quite small. That's because the active layer is water permeable. So this is why, yeah, the water flux and the salt rejection is actually very resilient against defects. You can puncture a membrane quite significantly, and still the salt rejection and the water permeability will not change significantly.
And you have an idea on how far you can push this? How many percentage of the membrane active surface can be damaged before you will see huge losses in rejection?
Yeah, actually quite far. So I did some sensitivity analysis with, of course, quite a few assumptions in there. But yeah, you can probably go up to about 1 in 10,000 of the area of the membrane being defected before you get really significant decreases in salt rejection.
Interesting. Good. I think it's only suited that we wish you the best of luck with with your further investigation and your further project. I would like to thank the speakers once again for their sharing their knowledge. Also, thank you to the people following this webinar, and we were happy to see you back on the 19th of May, where KULeuven and VITO will enlighten us with some of their projects. See you next time, and bye bye for now.